these are polymorphisms here, different kinds. And the number of alleles. Mostly SNPs and simple polymorphisms involve only 2 alleles. Whereas all these other ones like VNTRs and CNPs, you can have more than 2 alleles. Okay? So multi-allele can be documented by polymorphism. So here is an example of a microsatellite monitor. A microsatellite means these are 2 nucleotides, 3 and 4 nucleotides, tandem repeats. Like for example, AC, 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 like that. There are 10 copies of ACs in one area. So in some other instance, there could be 18 copies of AC, AC. In the same locus, this particular allele has 10, that has got 18. In a similar way, here is a 10 and 14 and 18 and so on. So by looking at these, see this is inherited here, these 2 are there, and those 2 therefore that is common between 2 individuals, and this allele is common between 2 individuals. Whereas if you look at each individual varies in this tandem repeats. Okay? Although there are 2 individuals having some commonality. And these things obviously are codominant segregation, like in the sense, this allele obviously is located at the same place, therefore they travel together essentially. It's a codominant kind of situation. Okay? Like codominance is a Mendelian inheritance. I'm sure you studied pea plants have got the pea shape as well as the tallness, both of them put together. It's a codominance, 2 inheritance factors. And the variable number of tandem repeats. These are slightly larger than those simple polymorphisms. And this allele 1 has got several repeats, allele 2 has got 4, and so on and so forth. And therefore, again, as you can see, you can mark these variable number of tandem repeats by this amplification and Southern blot or whatever, and you can detect those DNA fingerprinting. Fingerprinting is on a larger scale. That's the fingerprinting. It's not just only a small location where you can take 4 or 5 variable number of tandem repeats and analyze one individual. But here, you are going to do a tremendous number of VNTRs by simply probing it, by running either a Southern blot or by doing multiplex PCR nowadays. You could get large number of bands of being amplified. Look at here. These are monozygotic twins, and these 2 twins are essentially identical. Whereas in this place there is a variation, slight variation. These 2 are identical. So you can take a look at this kind of a pattern and tell the differences between individuals very easily. So if a person has committed a crime and you run this fingerprint, that fingerprint is going to be unique to the individual because it's going to be analyzing multiple times. Okay? In fact, in the olden days, like about what, this Southern is the one who really established this kind of fingerprinting a long time ago. And when these techniques were being established at one time, in fact, they were paying quite a bit. They actually called me one time to the court to identify whether this individual is having a similar fingerprint. Only nowadays everybody does DNA, but in those days only a few of us were doing this kind of analysis. But you don't want to go there because even though they pay $2,000 for 1 hour of going and testifying it, I'm afraid of going there. I refuse to go to the court because we don't know what will happen. A criminal can come after you sometime, right? So I just will never go to the court. There is another professor here, Robert Benjamin goes to court all the time. So, the inherited variation in polymorphisms in proteins. So, this is not polymorphisms nowadays after the human genome sequences come. The polymorphism studies have started long time ago, classically in blood groups. I'm sure everybody knows about blood groups, okay? And major histocompatibility complex. These 2 have been there for a long period of time in the literature. What are blood groups? We have different systems of blood groups. This one you have to study because if you study human genetics and say, I do not know about blood groups, it's not good. Therefore you have to, although everybody knows ABO, and one is compatible with the other, but what's the mechanism? How does it happen? You need to know a little bit about that. We have RH system and an ABO system. See, red cell has got such a has a B antigen but an antibody to A. And type AB has obviously both antigens but neither antibody. Okay. And type O has neither antigens but has an anti-A and anti-B. Because of that, you see, so if you take an O group and put an anti-A reaction with anti-A, it will be negative because it has an anti-other one. So the same thing here, anti-B is negative because the antibodies in the serum are anti-A and anti-B. So therefore there is no question of reaction of anti-A or anti-B because it already exists. Whereas if you take a group of A, you got a reaction with anti-A because there is an anti-B antibody because this is a group A. Group A has got an antigen but an anti-B antibody but it is an antigen A. Similar way, B group is negative and positive, and that is the reason why they type you for blood groups by looking at agglutination between the two blood groups. But what is this antigen? See, what happens is that there is a H antigen is there on the red cell surface. And a B allele, there's another allele which codes for an enzyme which adds D-galactose. Whereas A adds not the glucose, there is a glycosyl transferase. Whereas A allele adds acetyl-galactosamine. Both are adding to the surface antigen. One adds the galactose, another one adds an acetyl-galactosamine. But these proteins are slightly polymorphic. They are similar, but they have slightly different functions in terms of what moiety that is adding to the H antigen. And that is where it differs from A to B. Okay? So O group is due to a single base pair deletion. So because of these molecular mechanisms, now we know the molecular mechanisms. Previously they did not know, and they were only looking at these ABO blood groups for blood transfusion. If A use A, there's no big deal. B use B, there's no big deal. But O has been the universal donor, but still there are some issues with O. Okay? Whereas there is one more blood group called rhesus RH factor. It's coming from rhesus monkeys, that's why it's called RH factor. The RH factor is very unique and interesting. Suppose that's what is RHD antigen. Okay, there's an antigen, it's a protein encoded by chromosome 1. If the RHD is negative, okay, that is RH negative, there can be RH positive, that is the protein is present, RH negative is not present. So if there is an RH negative, if it is exposed to RH positive cells, they can develop an antibody. Okay? So let us say that RH negative mother marries an RH positive guy. And let us say the fetus, that is the baby, developing baby, is going to have RH positive cells. So what happens is the RH positive cells can cross the placental barrier and go to the mother's blood. Because mother has no RH positive at all. She's totally RH negative. But she sees now the cells are RH positive. So immediately there is going to be an immunological reaction. And thus she develops an antibody to RH positive cells. But these RH positive cells are creating an antibody in the mother's blood because she has an immunological defense. The fetus doesn't have immunological defense because the fetus don't have immunological defense at all. So mother develops antibodies, and those antibodies can travel back to the fetus. When it travels back to the fetus, because it is an antibody against its own cells, it is going to cause what is it called hemolysis. Hemolysis means the blood cells will lyse. That is called hemolytic disease of the newborn. So, in general, let us say that the mother loses the first child by some kind of an abortion or whatever. But she has already developed antibodies, correct? And therefore, in a second pregnancy, it doesn't matter. She's already having antibodies. They will go even early on, and therefore it will kill the second fetus. Therefore, the second pregnancy is more complicated also. Unfortunately, because they realize this significance, it's very easy for them to develop treatment. Because what they do is, when the mothers are around 22 weeks of pregnancy or whatever, they are going, or 28 weeks, what they are going to do is to go ahead and give her an immunoglobulin fraction of the so-called injection, which will neutralize the positive cells that are coming into the mother. Okay? So nowadays the RH is not a big deal, but in the olden days it was a big deal, because now we know the mechanism, so the treatment has come out for this particular disorder. There is one more complex, which is the major histocompatibility complex. Now, I was fortunate, I was in a laboratory when they cloned the very first gene of the major histocompatibility complex in 1879. So in this, there are called class 1 and class 2 antigens. Okay. The class 1 has got 3 loci, C, class 2 has got these 3 loci. But don't worry about the number of the loci, but what we need to worry about is the polymorphic variations in each locus. Okay. And this is enormous. So within this MHC locus also, there are multiple genes as well. So it is just a lot of genes of MHC. But in between, intergenic between MHC1, let's say A, B, and C, whatever number, you've got other genes as well. Like for example, one of the clotting factors, like factor 13 is present in major histocompatibility complex locus. Like this particular example. Here is a class 2 molecule, and those are the class 1. These are all class 2. These are all the number of loci, and these are the number of the genes there. But within this cluster also, there are a lot of other genes. That's what he's talking about. Okay? So obviously, if there is a polymorphism in this area, if there is a haplotype in this area, they will definitely be linked to these things. There is no question. Okay? Any disorder that is coming from this area, let us say 21 hydroxylase deficiency. If there is a polymorphism like one of these haplotypes, that will be definitely linked because they are right there within the locus. Okay? So imagine this way: if you have a bunch of cards and you shuffle them, okay, therefore they are random distribution. This is what happens in meiosis, right? Random recombination. But if I put a glue to 2 cards, in between 2 cards I put a glue. Whatever I shuffle, they're going to go together. So if the locus is very close to whatever the polymorphisms are, they are going to have tremendous association, like 100% association. Whereas if it is far away in a different place, it's going to have association. Okay? So we're going to talk more about